Cannabis nutrient elements. The first of cannabis macronutrient elements is nitrogen. Nitrogen stands as a fundamental element in cannabis cultivation, influencing crucial aspects of plant development. This essential nutrient is a primary component of chlorophyll, the green pigment responsible for photosynthesis. During the vegetative stage, cannabis plants have a heightened demand for nitrogen, promoting lush foliage and robust stems. Adequate nitrogen levels contribute to overall plant vigor, enhancing the plant's ability to capture and convert light into energy. However, precision is key in nitrogen management. While nitrogen is vital for vegetative growth, an excess during the flowering stage can lead to issues. Second is phosphorus. Phosphorus emerges as a critical nutrient in the realm of cannabis cultivation, wielding a profound influence on the plant's energy transfer, root development, and reproductive processes. As an essential component of ATP, or adenosine triphosphate, the primary energy currency in cells, phosphorus is indispensable for powering various metabolic reactions crucial for overall plant vitality. During the flowering stage, phosphorus takes center stage, promoting the formation of robust root systems and facilitating the synthesis of DNA and RNA, essential for optimal flower and seed development. Its role in energy storage and transfer also contributes to the efficient conversion of sunlight into chemical energy, enhancing the plant's ability to promote and accumulate cannabinoids. In cannabis cultivation, a balanced and timely supply of phosphorus is key to unlocking the plant's full potential, ensuring not only vigorous growth, but also bountiful and high-quality yields. Potassium emerges as a vital player in the orchestration of cannabis cultivation, exerting multifaceted effects on plant health, stress resistance, and overall yield. As a key regulator of water uptake and movement within plant cells, potassium aids in maintaining optimal turgor pressure, enhancing the plant's ability to withstand environmental fluctuations and resist diseases. Beyond its role in osmoregulation, potassium is instrumental in enzyme activation, particularly those involved in photosynthesis and energy metabolism. This nutrient fosters robust flower development, contributing to increased bud density and weight. Additionally, potassium plays a pivotal role in nutrient transport, ensuring the efficient movement of essential elements throughout the plant. Mastering potassium management in cannabis cultivation is therefore integral, as it not only supports plant vigor and resilience, but also plays a crucial role in maximizing the quality and quantity of cannabinoid production. Nitrogen's significance in cannabis cultivation extends far beyond being a mere component of amino acids and proteins. During the vegetative phase, when cannabis plants focus on developing their structural framework and lush foliage, a sufficient supply of nitrogen is essential. This nutrient facilitates the synthesis of chlorophyll, the green pigment responsible for capturing sunlight during photosynthesis. As a result, nitrogen directly influences the plant's photosynthetic capacity, ensuring efficient conversion of light energy into chemical energy. Robust nitrogen availability during this stage contributes to the formation of healthy, vibrant leaves, enabling the plant to maximize its potential for energy production. Moreover, nitrogen is integral to various metabolic processes within the cannabis plant. It actively participates in the formation of nucleic acids, the building blocks of DNA and RNA, crucial for the plant's genetic expression and overall growth. Nitrogen's involvement in enzyme activation further supports numerous biochemical reactions, ensuring that essential processes such as nutrient uptake, energy transfer, and hormone synthesis proceed optimally. This multifaceted role highlights nitrogen as a key driver behind the plant's metabolic machinery, orchestrating the intricacies of growth and development. While nitrogen is undeniably beneficial during the vegetative phase, its role must be carefully managed during the flowering stage. Excessive nitrogen levels during this critical period can lead to undesirable effects, such as delayed flowering, extended internodal spacing, and diminished flower development. Therefore, cultivators often adjust nutrient ratios to reduce nitrogen and encourage a seamless transition into the flowering stage, striking a delicate balance that promotes robust budding without compromising overall plant health. In summary, nitrogen's importance in cannabis cultivation extends from its foundational role in structural development and photosynthesis during the vegetative phase to its involvement in critical metabolic processes throughout the plant's life cycle. Cultivators must navigate the dynamic requirements of nitrogen, adjusting nutrient formulations to meet the changing needs of the cannabis plant as it progresses through different growth stages, ultimately ensuring optimal health, vigor, and cannabinoid production. Phosphorus stands as a cornerstone nutrient in cannabis cultivation, wielding a profound influence on the plant's ability to harness energy, establish robust root systems, and realize its full reproductive potential. As a structural component of ATP, or adenosine triphosphate, phosphorus plays a central role in energy transfer within plant cells. This fundamental process fuels various metabolic reactions critical for growth, development, and the synthesis of essential biomolecules. During the flowering stage, phosphorus becomes especially pivotal, driving the energy-intensive processes associated with flower and seed production. Adequate phosphorus availability is crucial for maximizing the plant's genetic potential during this critical phase. Beyond its energy-related functions, phosphorus is instrumental in promoting healthy root development. A well-established root system is essential for efficient nutrient uptake and water absorption, providing the plant with the necessary resources for sustained growth. Phosphorus facilitates the formation of nucleic acids, DNA, and RNA, supporting the plant's genetic expression and contributing to the development of strong, resilient roots. This robust root network not only anchors the plant securely in the substrate, but also enhances its capacity to explore and exploit the surrounding soil for nutrients. Phosphorus's influence on flowering extends beyond energy transfer and root development. It plays a key role in the synthesis of crucial biomolecules. As an essential component of nucleic acids, phosphorus contributes to the genetic material that directs the plant's growth and development. Additionally, phosphorus is a vital player in the formation of adenosine diphosphate, or ADP, and adenosine monophosphate, or AMP, molecules involved in energy storage and release. These energy transfer processes are particularly significant during the flowering phase when the plant diverts resources toward the production of flowers and, ultimately, cannabinoid-rich buds. Cultivators must carefully manage phosphorus levels to optimize cannabis growth. While a deficiency can lead to stunted growth and poor flower development, an excess may result in nutrient imbalances and potential environmental concerns. Striking the right balance, particularly during the flowering stage, is critical for achieving a harmonious interplay of energy transfer, root development, and genetic expression that culminates in the production of high-quality cannabis flowers. Potassium holds a pivotal role in cannabis cultivation, exerting a broad spectrum of effects that influence plant health, stress resilience, and overall crop yield. As a primary regulator of water movement within plant cells, potassium helps maintain optimal turgor pressure, enhancing the plant's ability to withstand environmental fluctuations such as drought or sudden changes in humidity. This crucial function contributes to the plant's overall stress resistance, bolstering its ability to cope with challenging growing conditions and minimizing the risk of diseases. In addition to its role in osmoregulation, potassium is an essential cofactor for numerous enzymes involved in photosynthesis and energy metabolism. By supporting these fundamental processes, potassium enhances the plant's capacity to convert sunlight into chemical energy, fostering robust growth and development. During the flowering stage, potassium becomes particularly crucial, influencing the synthesis and translocation of carbohydrates. This has a direct impact on flower development, promoting increased bud density and weight, ultimately contributing to higher yields of cannabinoid-rich flowers. Potassium's significance extends to nutrient transport within the plant. It plays a vital role in the movement of essential elements from the soil to various plant tissues, ensuring that nutrients are distributed efficiently. This aspect is essential for maintaining overall plant health and optimizing the availability of nutrients for key metabolic processes. Proper potassium management is therefore crucial for sustaining a balanced nutrient profile and preventing deficiencies or excesses that could impede cannabis plant growth. Cultivators must pay close attention to potassium levels throughout the cultivation cycle, tailoring nutrient formulations to the specific needs of the plant during different growth stages. By doing so, they can harness potassium's multifaceted contributions to support robust vegetative growth, enhance flowering, and maximize the quality and quantity of cannabinoid production, ultimately leading to a successful cannabis harvest. Cannabis micronutrient elements. calcium, boron, chlorine, iron, copper, manganese, zinc, sulfur, magnesium, and molybdenum. Calcium, despite being classified as a micronutrient, plays a macro role in cannabis cultivation, influencing various physiological processes that contribute to plant health, structure, and overall crop quality. One of the primary functions of calcium is its pivotal role in cell wall formation and integrity. As a structural component, calcium strengthens cell walls, promoting sturdy plant architecture and resilience. This is especially crucial during the vegetative phase when rapid cell division and elongation occur, providing cannabis plants with the structural support needed to withstand environmental stresses. Beyond its structural role, calcium is a key regulator of cell division and growth. It participates in essential enzymatic activities, facilitating the conversion of precursor molecules into complex compounds necessary for plant development. Additionally, calcium is involved in the activation of various enzymes that regulate nutrient uptake and transport within the plant. This ensures that essential elements are efficiently absorbed from the soil and transported to different parts of the cannabis plant, contributing to overall nutrient balance and health. Calcium also plays a critical role in mitigating various physiological disorders, such as blossom end rot. By maintaining a sufficient supply of calcium, cultivators can prevent the development of these disorders, ensuring optimal flower and fruit development. Moreover, calcium contributes to the plant's response to environmental stress, acting as a signaling molecule that helps activate stress-related pathways and enhance the plant's ability to cope with adverse conditions. Calcium is a micro but mighty nutrient in cannabis cultivation, supporting plant structure, growth, and stress resistance. Its influence extends from cell wall fortification to enzymatic regulation and stress response mechanisms, making it an indispensable element for cultivating robust and healthy cannabis plants. Proper management of calcium levels is essential for achieving optimal growth, reducing the risk of disorders, and ultimately maximizing the yield and quality of cannabis crops. Magnesium, a crucial micronutrient in cannabis cultivation, serves as the central component of the chlorophyll molecule, making it indispensable for photosynthesis. As an essential process driving the conversion of light energy into chemical energy, photosynthesis is foundational to plant growth. Magnesium's presence within the chlorophyll structure allows cannabis plants to capture sunlight effectively, facilitating the synthesis of carbohydrates and other organic compounds vital for energy storage and metabolic functions. Beyond its role in photosynthesis, magnesium is an activator for numerous enzymes involved in fundamental metabolic pathways. Enzymes that facilitate the conversion of phosphate compounds and the synthesis of nucleic acids rely on magnesium for optimal activity. This micronutrient's involvement in these processes contributes to the overall growth, development, and genetic expression of cannabis plants. Adequate magnesium levels are particularly crucial during periods of rapid growth, such as the vegetative phase, to support the plant's energy demands. Magnesium also plays a key role in nutrient uptake and transport within the plant. It influences the opening and closing of stomata, regulating gas exchange and water vapor loss. This in turn affects the absorption of essential nutrients from the soil. Proper magnesium levels contribute to a balanced nutrient profile, preventing deficiencies and optimizing the plant's ability to access other vital elements necessary for healthy development. In addition to its metabolic and regulatory functions, magnesium contributes to stress resistance in cannabis plants. It acts as a cofactor for enzymes involved in antioxidant defense mechanisms, helping the plant combat oxidative stress caused by environmental factors such as high light intensity or extreme temperatures. As a result, maintaining adequate magnesium levels is crucial for cultivating resilient cannabis plants capable of thriving in varying growing conditions. Sulfur, often regarded as a secondary but vital micronutrient in cannabis cultivation, plays a diverse and essential role in the overall health and development of cannabis plants. Sulfur is a key constituent of amino acids, which are the building blocks of proteins. Proteins, in turn, are critical for numerous physiological functions within the plant, including enzyme activity, structural support, and the synthesis of essential compounds. Therefore, a sufficient supply of sulfur ensures the production of high-quality proteins, contributing to robust growth and development. Sulfur's involvement extends to the synthesis of vitamins and coenzymes within the cannabis plant. It plays a crucial role in the formation of thiamine and biotin, essential compounds that participate in various metabolic processes, including energy transfer and the breakdown of carbohydrates. This underscores the importance of sulfur in supporting the plant's overall metabolic activity, ensuring efficient nutrient utilization and energy production. Additionally, sulfur contributes to the production of secondary metabolites in cannabis, such as certain terpenes and defense compounds. These secondary metabolites play a role in the plant's interactions with its environment, including defense against pests and pathogens. By promoting the synthesis of these compounds, sulfur enhances the plant's natural defense mechanisms, contributing to its resilience in the face of environmental stresses. Sulfur's influence on pH regulation in the soil is noteworthy. It can contribute to soil acidity when needed, which is particularly beneficial in mitigating alkaline conditions that may hinder the availability of other essential nutrients. This emphasizes the importance of sulfur not only as a direct contributor to plant nutrition, but also as a factor that indirectly influences the overall nutrient availability in the growing medium. Boron, often considered a trace element in cannabis cultivation, plays a vital role in various physiological processes that are essential for the overall health and development of cannabis plants. One of its primary functions is in cell wall formation and stability. Boron is integral to the synthesis of pectin, a component of plant cell wall. Proper cell wall structure is crucial for maintaining cell integrity, supporting structural stability, and facilitating essential functions like nutrient transport. Boron also influences reproductive processes in cannabis, particularly during flower and seed development. It is involved in the formation of pollen tubes, facilitating the transportation of male gametes to the female reproductive organs. This process is fundamental for successful pollination and subsequent seed formation. Adequate boron levels contribute to optimal fertility and seed set, crucial considerations for cultivators aiming to produce viable and genetically stable seeds. Furthermore, boron plays a vital role in the metabolism of carbohydrates and the regulation of plant hormones. It is involved in the synthesis of sugars and the transport of carbohydrates within the plant. Additionally, boron influences the activity of various enzymes related to hormone metabolism, including auxins, gibberellins, and cytokinins. This makes boron a key player in regulating processes such as cell elongation, flowering, and fruit development. Despite its importance, boron availability must be carefully managed, as excess boron can be detrimental to cannabis plants. Boron toxicity can lead to symptoms such as leaf necrosis and impaired growth. Therefore, maintaining a balanced and appropriate level of boron is crucial for harnessing its positive effects on cell structure, reproductive processes, and hormonal regulation without causing adverse effects. Manganese, a micronutrient essential for cannabis cultivation, plays a vital role in various metabolic and enzymatic processes crucial for plant growth and development. One of its primary functions is in photosynthesis, where manganese is a critical component of the water-splitting complex in photosystem 2. This complex is essential for the conversion of light energy into chemical energy, facilitating the production of oxygen and energy-rich compounds. Adequate manganese levels ensure the efficiency of photosynthesis, promoting the synthesis of carbohydrates that fuel the plant's growth. Manganese is also involved in the activation of enzymes responsible for antioxidant defense mechanisms. These enzymes, such as superoxide dismutase, help the plant mitigate oxidative stress by neutralizing harmful reactive oxygen species. By playing a role in the plant's antioxidant system, manganese contributes to the overall stress tolerance of cannabis plants, enhancing their ability to withstand environmental challenges, including fluctuations in light intensity and temperature. Furthermore, manganese participates in the metabolism of nitrogen and the synthesis of amino acids, proteins, and nucleic acids. This involvement in nitrogen metabolism contributes to the efficient utilization of nitrogen by the plant, optimizing nutrient uptake and assimilation. The synthesis of essential biomolecules, including proteins and nucleic acids, is crucial for various cellular functions, including growth, reproduction, and genetic expression. Therefore, maintaining proper manganese levels is integral to supporting these fundamental processes in cannabis plants. However, like other micronutrients, manganese must be carefully managed to prevent toxicity. Excess manganese can lead to symptoms such as leaf discoloration, reduced growth, and impaired nutrient uptake. Therefore, cultivators should monitor manganese levels closely, ensuring they fall within the optimal range to promote healthy growth and metabolic processes in cannabis plants. Zinc, a micronutrient essential for cannabis cultivation, plays a crucial role in various biochemical processes that are fundamental to plant growth, development, and overall health. One of its primary functions is as a cofactor for numerous enzymes involved in the synthesis of nucleic acids and proteins. Zinc is a vital component of DNA and RNA polymerases, the enzymes responsible for the replication and transcription of genetic material. As a result, zinc contributes directly to the plant's genetic expression, growth, and development. Additionally, zinc is integral to the activation of enzymes involved in auxin synthesis and metabolism. Auxins are key plant hormones that regulate processes such as cell elongation, root development, and flowering. By influencing auxin-related enzymes, zinc plays a crucial role in shaping the plant's architecture, root system, and reproductive processes. This makes zinc indispensable for cultivators seeking to optimize plant structure and maximize yields. Zinc is also involved in the formation of chlorophyll, the green pigment responsible for photosynthesis. As a constituent of chlorophyll-binding proteins, zinc ensures the efficient capture of light energy during photosynthesis, promoting the synthesis of carbohydrates that fuel the plant's growth. Moreover, zinc plays a role in the activation of enzymes involved in the metabolism of carbohydrates and other energy-related processes. This highlights zinc's multifaceted contributions to energy transfer and storage within the plant. While zinc is essential for cannabis plants, maintaining the right balance is critical. Zinc deficiencies or excesses can lead to visible symptoms such as stunted growth, leaf discoloration, and impaired flowering. Therefore, careful monitoring of zinc levels and providing a balanced nutrient solution are crucial for cannabis cultivators to ensure optimal plant nutrition, support metabolic processes, and achieve healthy and vigorous plant development. Chlorine, while not typically considered a micronutrient for most plants, is still relevant in cannabis cultivation as it plays a role in various physiological processes. Chlorine's primary function is associated with photosynthesis, where it participates in the water-splitting reaction during photosystem 2. This reaction is crucial for the generation of oxygen and the formation of energy-rich compounds, contributing directly to the plant's energy production. While chlorine is often obtained from water sources, it's worth noting that excessive levels can be harmful. Therefore, maintaining an appropriate balance is essential for optimal photosynthetic efficiency. In addition to its role in photosynthesis, chlorine is involved in the regulation of stomatal movement. Stomata are tiny pores on the plant's surface that control gas exchange and water vapor loss. Chlorine influences the osmotic potential within guard cells, affecting the opening and closing of stomata. Proper stomatal regulation is essential for the plant to balance gas exchange and water conservation, particularly in response to environmental factors such as humidity and temperature. This underscores the importance of chlorine in supporting the plant's adaptation to various environmental conditions. Chlorine also participates in the synthesis of organic molecules, including amino acids and proteins. It acts as a cofactor for enzymes involved in nitrogen metabolism and amino acid synthesis, contributing to the plant's overall protein content. Protein synthesis is fundamental for various cellular functions, including growth, reproduction, and stress response. Thus, maintaining an adequate supply of chlorine is important for supporting these essential metabolic processes in cannabis plants. While chlorine deficiencies are rare in cannabis cultivation due to its presence in water sources, excessive levels can lead to toxicity and negatively impact plant health. Therefore, cultivators should be mindful of chlorine levels in their growing media and water sources, ensuring a balanced nutrient profile to promote healthy growth and optimize the plant's metabolic functions. In summary, while not a traditional micronutrient, chlorine plays a nuanced role in cannabis cultivation, contributing to photosynthesis, stomatal regulation, and essential metabolic processes. Balancing chlorine levels is crucial for harnessing its benefits without causing harm to the plants. Molybdenum, despite being required by plants in trace amounts, is a crucial micronutrient in cannabis cultivation due to its involvement in key enzymatic processes. Molybdenum is an essential component of enzymes such as nitrogenase and nitrate reductase, which are pivotal for nitrogen metabolism. Nitrogenase is responsible for converting atmospheric nitrogen into a form that plants can utilize, promoting healthy growth. Nitrate reductase, on the other hand, facilitates the conversion of nitrate into ammonia during nitrogen assimilation. This process is vital for the synthesis of amino acids, proteins, and other nitrogen-containing compounds essential for plant development. Molybdenum's significance extends to its role in the activation of enzymes involved in sulfur metabolism. It is a cofactor for sulfate oxidase, an enzyme responsible for converting sulfite to sulfate. Sulfate is crucial for the synthesis of sulfur-containing amino acids, such as cysteine and methionine, which are essential building blocks for proteins. Therefore, molybdenum contributes to the overall sulfur nutrition of cannabis plants, supporting the synthesis of important biomolecules. Molybdenum's impact on iron uptake and utilization is noteworthy. It is involved in the conversion of iron into a form that can be easily transported within the plant. This process is vital for maintaining optimal iron levels, which in turn influences chlorophyll synthesis and energy production. By facilitating iron metabolism, molybdenum ensures that cannabis plants can efficiently utilize this essential nutrient for photosynthesis and other metabolic functions. While molybdenum deficiencies are rare, they can manifest as symptoms such as yellowing of older leaves and stunted growth. Therefore, ensuring an adequate supply of molybdenum in the nutrient solution is crucial for preventing deficiencies and optimizing the plant's ability to carry out essential metabolic processes. In summary, molybdenum's role in nitrogen and sulfur metabolism, as well as its impact on iron utilization, highlight its importance as a micronutrient in cannabis cultivation. Proper management of molybdenum levels is integral to supporting robust growth, nutrient assimilation, and overall crop quality. Copper, classified as a micronutrient, plays a critical role in various biochemical processes that contribute to the overall health and development of cannabis plants. One of its primary functions is its involvement in photosynthesis. Copper acts as a cofactor for plastocyanin, an essential protein that facilitates the transfer of electrons during the light-dependent reactions of photosynthesis. This electron transfer is crucial for the generation of energy-rich compounds and the production of oxygen, ultimately supporting the plant's energy needs. Copper is also an essential component of enzymes involved in redox reactions, which play a vital role in cellular respiration. Enzymes such as cytochrome C oxidase, which is part of the electron transport chain in the mitochondria, require copper for their activity. This underscores copper's importance in the plant's energy metabolism, ensuring efficient energy production and utilization. Moreover, copper is integral to the formation and stabilization of lignin, a complex polymer that provides structural support to plant cell walls. By contributing to lignin synthesis, copper enhances the plant's ability to withstand environmental stresses and pathogens, promoting overall structural integrity. This is particularly relevant in cannabis cultivation, where sturdy plant structure is essential for supporting heavy buds during the flowering stage. While copper is essential, excessive levels can be toxic to plants. Therefore, maintaining a balanced supply of copper is crucial for preventing toxicity, while ensuring the plant receives sufficient amounts for its metabolic processes. Copper deficiencies can manifest as distorted leaf growth and chlorosis, highlighting the importance of careful nutrient management in cannabis cultivation. In summary, copper's pivotal roles in photosynthesis, cellular respiration, and structural support emphasize its significance as a micronutrient in cannabis cultivation. Proper management of copper levels is essential for supporting robust growth, metabolic processes, and overall crop quality. Iron, a crucial micronutrient in cannabis cultivation, is essential for various physiological processes that contribute to plant growth and development. One of its primary functions is in chlorophyll synthesis, the green pigment responsible for photosynthesis. Iron is a key component of the heme group within chlorophyll molecules, facilitating the capture and conversion of light energy into chemical energy. Adequate iron levels are essential for sustaining optimal photosynthetic activity, promoting the synthesis of carbohydrates that fuel the plant's growth. Beyond its role in photosynthesis, iron is involved in the formation of enzymes and proteins crucial for metabolic processes. Iron acts as a cofactor for enzymes involved in the synthesis of DNA, RNA, and other essential biomolecules. This participation in nucleic acid metabolism contributes to genetic expression, supporting the plant's growth, reproduction, and overall physiological functions. Iron plays a pivotal role in the electron transport chain during cellular respiration. It acts as a cofactor for enzymes involved in the transfer of electrons within the mitochondria, facilitating the production of adenosine triphosphate, or ATP, the primary energy currency of the cell. This underscores iron's significance in energy metabolism, ensuring efficient energy production and utilization by the cannabis plant. While iron is essential, its availability can be influenced by factors such as pH levels in the growing medium. Iron uptake by cannabis plants is optimal within a specific pH range, and deviations can lead to deficiencies or toxicities. Iron deficiencies manifest as interveinal chlorosis, where the veins of the leaves remain green while the tissue between them turns yellow. Therefore, maintaining appropriate pH levels and providing a balanced nutrient solution are crucial for ensuring that cannabis plants receive sufficient iron to support their metabolic functions and overall health. In summary, iron's importance as a micronutrient in cannabis cultivation extends from its role in chlorophyll synthesis and photosynthesis to its involvement in genetic expression and energy metabolism. Proper management of iron levels is fundamental for promoting robust growth, optimizing metabolic processes, and ensuring the overall vitality of cannabis crops. Cultivating terroir. Delve into the realm of commercial cannabis cultivation and discover the artistic narrative behind Cultivating Terroir, found only at www.smokybee420.com.